Hi, my name is Alexandra, and for my project with Talaria, I did a comparative analysis on the orthologs of Gossypium hirsutum, which is basically just upland cotton. And I did a comparative analysis of the Rab3GAP gene through multiple sequence alignments. So essentially, RABs are a family of small GTP binding proteins that are part of the Ras superfamily, and they play a crucial role in regulating intracellular vesicle trafficking by cycling between inactive GDP-bound states and active GTP-bound states, as seen in the diagram I present here. This cycle essentially controls various aspects of membrane trafficking within the cell, and in order for it to work at an accelerated rate, there are certain molecules that help drive these processes to help them work faster. And these molecules are called Rab3GAPs, which is short for Rab-GTPase activating proteins. So these are essentially regulators that control the activity of Rab-GTPs, and they accelerate the conversion of the Rab-GTPases from their active GTP-bound state to their inactive GTP-bound state through promoting GTP hydrolysis, which is essentially the splitting of molecules by adding a molecule of water, and helping break down molecules so they can pass through cell walls and membranes. So Rab3GAPs are essential for proper intracellular vesicle trafficking, and they're involved in various cellular processes, both in animals and plants. Specifically, there's a subfamily of Rab3GAPs called Rab3GAPs in many organisms that specialize in stress response and in animals, neurotransmitter release in neurons. However, the reason we're studying this in cotton is because in cotton, the function of Rab3GAPs is unknown, and it's not studied enough to the point that it is in other organisms. So in this study, we're looking at Rab3GAPs, and we investigated orthologues of upland cotton. So orthologues are essentially different expressions of a gene throughout different organisms. So they are homologous sequences that are expressed in different organisms, and they have the same ancestral lineage. So it's a good way to compare how different organisms have gained a certain gene or sequence and how this function has essentially evolved over time to become what it is in that certain organism. So doing this, we wanted to know which subfamily was more like the human family, the human Rab3GAP, and what might this mean for its function in cotton. So we also wanted to know, sorry, let me switch. So essentially our research questions were to know what we wanted to know, which cotton subfamily was most resemblant of the animal lineage, and what might that mean for the function of Rab3GAP in cotton. We also wanted to know if any catalytic residues from animal sequences were conserved in the plants, and if so, which ones and why might they be important. Additionally, we wanted to see which similarities there were between subfamilies of the Rab3GTPase activating catalytic subunit, which is the subunit that this Rab3GAP family of cotton falls into. So the method we used was multiple sequence alignments using the sequences we found in a phylogenetic tree. So multiple sequence alignments align 3 or more sequences of genes, and through this process, they highlight the alignments and are used to infer evolutionary relationships between different sequences, and they are used to infer evolutionary relationships via phylogenetic analysis, which you can see through the diagram presented here. So essentially what we did is we explored a phylogenetic database, and we pruned the Rab3GAP subfamily, and we selected different organisms of interest that would display an interesting ancestor lineage, and that could convey a pattern of how this gene was passed on throughout different organisms. So if you look on the screen, you can see two subfamilies of this Rab3GAP gene distinguished throughout the plant sequences. And this essentially was done by including a series of different monocots and dicots throughout the pruning of the organisms. And this is able to show how different monocots or dicots have essentially taken this gene and how they've used it in their evolutionary history. And we also included an organism that was not a monocot or dicot, which was P. patens, because it's an important organism to understand how the Rab3GAP gene has diversified as a species, and grown more reproductively diverse and complex. So P. patens is a homospore. So essentially it is less evolved in structure and function than many of the more developed angiospores, which include monocots and dicots. So, and then we also included sequences of animal orthologues, so we can compare the human sequences to the plant sequences and see what similarities or differences there are between either subfamily. So in short, the methods that we used were, as I mentioned before, we created a phylogenetic tree using phylogenes, and these sequences were selected to include organisms of interest. And through this, using ID mapping, we were able to extract the sequences in a FASTA format, which is essentially a script of simple text that depicts the sequence of the protein that we're looking at. And it collects them in a batch order and essentially gives you a whole file of all the sequences that you're looking at. And then these sequences were aligned in a tool called Cluster Omega, which helps number the residues in order to prepare for multiple sequence analysis. And then ESPRIPT, which is a multiple sequence analysis tool, was used to render similarities in the secondary structure and sequence information from the aligned sequences from the cluster file. And this essentially would help us understand visually which residues were more conserved throughout all the subfamilies. And this can help us understand, okay, which pieces of these sequences are the most important in the functional and structural aspects of this protein, and how might they be important in the preservation of the function and the job of this molecule. And then also we used Chimera X, which is a molecular visualization tool, to compare the structural differences between subfamilies. And this was used essentially to correlate written sequences to physical structures in order to understand which areas display differences in structures based on sequences. And then lastly, we created a sequence logo using WebLogo, which is essentially a visual representation of the protein sequence, except various amino acids are more highlighted based on frequency of appearance. And this can be used to indicate higher conservation levels and therefore indicate which areas or motifs of the sequence are more preserved and should be paid more attention to. So as a result of the structure analysis using Chimera X, the subfamilies found in the phylogenetic tree are shown here. So if you can see my mouse, the figure under structure A is the subfamily MQJ, which was the first subfamily seen on the left-hand side of the phylogenetic tree. And as you can see, there is a extra domain that you can also see in the animal structure, but you cannot see in the MCO15 structure of figure B. So each of the A, B, and C structures have a common domain that is shaped similar to a V, and this is found all throughout each of these structures and is known as a common domain called the Rab3GAP catalytic subunit. However, interestingly, when we viewed the MQJ structure, there was an additional domain that was not assigned any official structure, and this presented a area of interest because the structure itself and its function was unknown, except as you can see, based on figure 3, it was also conserved in the human structure. So using different analysis tools, we were able to isolate the sequence segments that either of these domains in figure A and C are part of. And we noticed that they both have 2 chains in each of this sequences. So essentially, these 2 chains indicate that this sequence is better conserved evolutionarily throughout time because it attaches in multiple areas to the original conserved domain, and therefore it might be more structurally important, especially to the structure in figure A, because this is a structure we don't know much about, and using this information about the amount of chains and how many ties it has to the original domain can kind of hint that it is an important feature in the structure of this protein, and it has functional importance that might be worthwhile to study further. And then going on to the sequence aspect of the multiple sequence alignment, the following A, B, and C representations highlight the different multiple sequence analyses that we did through each subfamily. So these following sequences differ in each subfamily based on the phylogenetic tree. And as you can see, there are these things called motifs that are preserved throughout each of the sequences. So motifs are essentially small segments of sequences that show high preservation. And these areas are functionally important because specifically for a Rab3GAP, these are catalytic residues, which means that they have a functional importance in accelerating the rate of GTP hydrolysis, as mentioned before. So this is a motif mentioned, the IDR motif is a motif mentioned in multiple literature pieces, and it is useful because it helps accelerate catalysis. And as you can see throughout the annotations, it's conserved through all of the subfamilies. However, it is most conserved in subfamily MCO and the animal subfamily. So essentially, the reason for this, as we presume, is because of the similarities in the catalytic domain. And the reason it's preserved is because this is an essential portion of the protein that is used to accelerate these catalytic features of the Rab3GAP. So as I mentioned before, if you can view structure A, sorry, the MQJ family has an additional domain that is unknown in its function. If you can view the sequence here on subfamily MQJ, you can see that the first letter, the I in the IDR motif, is not as preserved as it is in the other subfamilies. Instead, it's replaced with a V, and this is essentially replacing isoleucine with valine, and there is a reason for this phenomenon. It's called conservative amino acids, and this typically happens when there are amino acids that are similar in function and structure, or they're similar in structure, and in turn they become similarly useful in function, and therefore, although the conservation of the original amino acid is not 100%, the structural and functional conservation is maintained by that replacement, because regardless of if the original amino acid is not conserved, the new one with similar structure can still fulfill those functional capabilities. And we believe, well, through the analysis, it's believed that possibly the additional domain of the MQJ subfamily might be able to help additionally support this motif, because even though it doesn't have full sequence conservation, it might have functional conservation through the help of that additional domain. And through analysis, we've kind of concluded that possibly that additional domain might be a supplemental structural feature that might help accelerate catalysis through helping this motif on the subfamily MQJ. Additionally, throughout the multiple sequence alignment, we were able to use the sequence logo as depicted above the sequences and the multiple sequence alignment itself to highlight areas of interest that might be useful for future analysis and developing new motifs. So new motifs experimentally. So as I mentioned before, motifs are areas of highly conserved residues that may indicate functional importance in catalytic activity or binding activity. So through the sequence logo and the multiple sequence alignment, identifying areas of high conservation, as you see through the coloration in these red letterings, as well as the height of the letters in the sequence logo, has kind of created a way to analyze which areas might be useful to revisit later in research to understand how they might be important to this protein specifically, and or this gene specifically. And through experimental research, we can kind of look at these more in depth and see if they have any catalytic properties, or they're just simply areas of high conservation that are structurally or functionally important to the protein. Excuse me. So as you can see, throughout the multiple sequence alignment, there are various segments that are highly conserved through the red coloration. And through that, I was able to collect 4 different sets of sequences and propose them for further research in investigating new motifs. So first of all, I proposed the PMLTED motif, the EDFRFSPDW motif, the HKGAP-S motif, and the ERWE motif. So I think given the results of the multiple sequence alignment, it would be valuable to investigate these areas specifically in further research to understand if they have catalytic properties that might be conserved throughout various subfamilies, and it might be nice to understand how they function in order to understand more about how the Rab3GAP in cotton itself functions. So in conclusion, the multiple sequence alignments emphasized the IDR motif as the best conserved motif throughout the Rab3GAP gene family, And although numerous residues displayed conservative variations, namely aspartic acid and I think aspartic acid displayed the highest degree of conservation across all the homologues, which was part of the IDR motif. And this might underscore its relevance across the Rab3 GAPs in accelerating the GAP's ability to regulate the Rab3 GTPases. Structurally, the MQJ subfamily is believed to be the most resemblant of the animal homologues due to the additional beta sheet structure represented in the family domain as shown before. And this is an unclassified domain, though all the animal homologues contain the structure in their predicted AlphaFold model. So therefore, speculation might arise that the homologous domain in the MQJ subfamily acts to strengthen the catalytic functions of the common domain by supporting the catalytic motifs in its core. Although the function of G. hirsutum Rab3GAP remains individually unknown, the knowledge about its lineage highlights its ancestors' property, which can allow for the basis of comparison when searching new information about this gene's identity as a Rab3GAP protein. The importance of studying cotton is that it is a major agricultural crop, and through genetic comparisons, we can uncover more information about its molecular mechanisms and especially about its functions in cotton. So thank you so much, and thank you to my mentor for helping me throughout this project. Thank you, Talaria, for giving me this opportunity. The following are my references, and if anyone has any questions, please let me know.